Good evening. My name is Johanna Key. My major is Food and Nutritional Science. My faculty advisor was Dr. Sharma, and I will be presenting an article entitled Short Root and Scarecrow Regulate Leaf Growth in Arabidopsis by Stimulating S-phase Progression of the Cell Cycle. So what is Arabidopsis thaliana? It is a small flowering plant that is widely used as a model organism for biological use. It is a member of the mustard family, which includes species such as radishes and cabbage, which are cultivated species. And it's not widely used in agriculture. However, it is very advantageous towards basic research regarding genetics and molecular biology. So plants generally have two systems. They have a shoot system and a root system. And so the shoot system will be everything that grows above ground. So this includes the stem, the appendages, the leaves, the buds, the flowers, all of that. And so with the shoot system, it produces sugar using photosynthesis, which is used for energy, and it also carries out reproduction for the plant. And then you have the root system, and this is what is under the soil. So it anchors the soil, the plant into the soil, as well as penetrating the soil and allowing the plant to absorb all of the nutrients and the water that it needs. And it also stores food. So shoot growth phenotype, this is basically the growth of the shoots as a result of the phenotype and how it reacts to the environment. So an example would be a plant that is growing in a location where there's not a lot of sunlight. This may impact the plant in a negative way. So it would impact the shoot growth in a way that the shoot wouldn't be as large as it would be in the sun. So stem cells are undifferentiated cells that can duplicate themselves, and they can also differentiate into other cells that basically build up our tissues and our organs and so on. And these cells are found in abundance in the root and shoot apical meristems of the plant. And the balance between the stem cell maintenance and differentiation of cells is what basically controls the root and shoot growth. So there were two genes that were really isolated in this article experiment. And these two genes were the short root gene and the scarecrow gene. And so both of these are members of the generally recognized as safe, also known as the grass family of transcription factors. And transcription factors are basically just the off switch for DNA. And then, so the short root and the scarecrow genes are required for maintenance of the stem cells in the Arabidopsis root meristem, which ensures plant growth. So when they are mutated, the Keynesian center is disoriented and the stem cell activity is disrupted, leading to a cessation in root growth. So this is just a recap of the phases of cell division. First, you're going to have your G phase, which is also known as your first gap phase. And this is basically when the cell prepares for division. In the S phase, this is where synthesis of the DNA is actually occurring, where the DNA is replicated, and then it leaves two sister chromatids. After that is G2 phase, which is also known as the gap2 phase. And this is where the cell prepares for mitosis and cytokinesis. And then following that is the M phase, where mitosis and cytokinesis actually takes place. And then you have your interphase, which is basically G phase, 1, 2, and S phase. So the objective of this experiment was to use the Arabidopsis leaf as a system to specifically study the function of the short root and scarecrow genes in cell cycle regulation, separate from their role in basically stem cell specification and how it relates to the shoot growth phenotype. So the materials and methods of this study, the plant materials and growth conditions. So the Arabidopsis plant was used, of course, to study the mutants shortroot 6, shortroot 3, scarecrow 3, and it also used promoter fusions to promote these genes. And then you have your seeds were, for in vitro analysis, were sterilized in 3% bleach, and then they were sown in a common medium used for plant growth. After that, root growth samples were placed into 1 gram of agar, and they were placed on square plates. And they were placed on square plates so that the root growth plates could be placed vertically instead of horizontally, so that the roots would be able to grow vertically, basically. And the horizontal plates were the leaf growth plates. So for growth analysis, the rosette growth was analyzed by taking pictures of the petri dishes, and these samples were followed for a total of 18 days. And then the total leaf blade area of plants 1 and 2 of 5 plants from day 25 to 24 were measured with dark field binoculars. And then they also underwent flow cytometry. The leaves were lacerated with a laser blade, and then they were suspended into 1.5 milliliters of buffer. And then the supernits were filtered off, and then the leaves were basically stained with a DNA stain, and it was a blue color. And then the nuclei were analyzed using cytometry. And so these are the results. This first image basically illustrates that the rosette growth is reduced in the short root and scarecrow mutants. So at the top is going to be your, basically it's just a picture analysis of the growth. So in the middle is the wild type plant, and that's the one that just grows in nature. At the on the left side, that's the short root plant, and as you can see, that one had the most inhibited growth. And then on the right, that's the scarecrow plant, and that one had inhibited growth, but not as much as the short root. Then part B, it was basically a growth analysis of watching the plants grow from 48 hours to 400 hours. And then at the bottom, they basically just took the leaves off the actual plants and lined them up so that you could really compare them. So at the top is your wild type, at the middle is your scarecrow, and at the bottom is your short root. So this picture basically shows that the shoot phenotype is not dependent on aberrant root phenotype. So the plant to the far left, this is basically a short root 3 mutant. And then the plant in the middle is part short root 3 and part reverted back to wild type. So the roots are reverted back to wild type, and the shoot is a short root 3. And then the last plant is just a complete reversion back to wild type. So as you can see, even though they reverted the roots of the plant in the middle back to wild type, and they're all strong and long, the actual shoot of the plant is still hindered. So it does not. So the shoot growth is not impacted by the root growth. And then this last picture basically shows the progression of the cell division gradient in short root and scarecrow mutants. And so it was followed. They followed it for a total of 3 days, and basically cell division stops in a tip to base gradient. So as you can see, for the short root, by day 10, it was already only a little bit of cell division left taking place. And by day 11 and 12, there was nothing taking place at all. So this just shows that short root mutants cause an early exit of the cell proliferating from the proliferating phase, which is the M phase. And so for the discussion, the results demonstrated that the inhibition of the leaf growth in short root and scarecrow mutants is not a secondary effect of the compromised root development, but it's actually an effect of basically cell division in the leaves. There was some hindered cell division, so of course there would be hindered growth in the leaves. And it also took place because of an early exit of the proliferation phase. And then in conclusion, in contrast to their specific roles in stem cell maintenance in the root, scarecrow gene and short root gene their primary function is as general regulators of cell proliferation in the leaves, and retarded leaf growth in scarecrow and short root mutants is not a secondary effect of the compromised root growth. It is independent, and it's caused by the loss of the short root and the scarecrow function in the tissue. And these are my references.